So now you've read the text, I'm going to go through it with you so that you gain a deeper understanding of what you're looking at. We're going to start with the eye and sight. So if we look at the picture of the eye, although there's a lot of information here, I'm going to give you a clear description of what you need to know. You need to have an understanding of how the cornea works, the lens, the pupil and the iris, the ciliary muscle, the retina, and the optic nerve. So these are the important ones, and the fovea, I might throw that in a bit later. Light travels through the cornea, through the pupil, through the lens, and focuses on the back of the eye at the retina. So that's what needs to happen. So let's look at those parts in detail. The cornea is just a protective layer around the outside of the eye's opening. It's transparent, and that's about it really. So the cornea is a transparent protective layer. The pupil and the iris, they work together. And if we look at somebody's eyes head on, it's probably what we're most familiar with. We have a circle of colour and a small black hole in the centre. Okay, so the iris is the colourful bit. I sometimes remember irises are flowers and flowers are colourful. I don't know, something simple like that. And the pupil. Okay, and that's in the centre. What are these for? Well, if we have a bright light, then there's too much light in the room, and then the iris closes up the pupil to give it a very small hole. So bright light, we have a very small pupil. But if it starts to get very dark, then the pupil needs to open up to let as much light in as possible. Okay, so you get a nice large pupil when it's very dim. So that's what the iris and the pupil are for. The lens focuses light, and we'll have a little look at that in class over the next few days. As light comes in, the light must focus through the lens, and depending on where the object is, the lens must actually change in size. How does it change in size? With the action of the ciliary muscles. Okay, so these will basically contract and relax to change the size of the lens thickness. Okay, so let's have a look at how light travels. Light will travel in. Okay, so I'm going to draw 3 lines there. 1, 2, 3. So light travels in through the pupil, past the cornea, which is transparent, in through the pupil, to the lens, and from the lens it focuses at the back of the retina. Now this is a simple representation of how it works. At the retina, light signals, we could say light energy, is converted into an electrical signal, and that's sent down the optic nerve to the brain. So the role of the optic nerve is to transfer that information from the retina to the brain. The fovea, here, this little dimple, I think you can see it there, happens to be the place which has the highest concentration of photoreceptors. By photoreceptors, these are photon receiving receptor cells. Okay, so these are the actual parts of the retina, the cells in the retina that receive that light information. At that point in the fovea, there is the highest concentration of them. So therefore, it's where you would get the most information and thus build the clearest picture. So that's the ideal place to focus a perfect image. I'm now going to explain depth of vision. And I'm going to use this diagram here to help explain it. By definition, the depth of vision is the range of object distances from the eye within which objects or points on an object can be seen clearly. So there is a range of space, you could say, where your eye is capable of focusing clear images on the retina. Okay, and how do we describe that area? Well, the closest possible point where we can still see a focused image is called the near point. Okay, so the near point is the closest possible image that we can still focus, and the far point is the furthest possible image. So that's how far something can be for you to still be able to focus upon it. In addition to this depth of vision idea, okay, which we have here, we have lens accommodation. Our ability to be able to change our focus from something that is close to something that is far is all thanks to the lens of the eye. The lens of the eye actually changes shape in order for us to be able to see things clearly depending on where they are. The basic idea is this. A fat lens, like this one, would have a very short focal length. Okay, oops. And that's the principal axis. Now, because it has such a short focal length, it can accommodate the focusing of objects that are very nearby. So when you look at something, let me rub that off because that's really messy. Okay, near. So when you have an object that's very close, your lens actually gets thicker. And how does that happen? Well, the ciliary muscles actually contract. In fact, what they do is they relax. And this is the lens in its relaxed shape. Okay. What happens when you then need to focus your vision onto something that's far? Well, then the ciliary muscles, they actually get really stretched and tight. And what they do is they pull the lens into this thinner shape. This thin lens has a really long focal length. And it allows the lens then to focus on objects that are very far away. And that's what we call accommodation. The lens in your eye changes shape in order to accommodate different distances of object. How does it do this? The ciliary muscles contract and relax to change the shape of the lens. Now I'm going to talk about the rods and the cones. These are photoreceptors which line the back of the retina. Okay, so they are basically the receptors that are most vital to generating an image. And rods and cones each generate what's called scotopic and photopic vision. Okay, so what you need to be able to do is explain what the distribution of rods and cones in the retina are and describe the difference between scotopic and photopic vision. Here I've highlighted the important points. Rods are sensitive to light of low intensity. So light does not have to be bright in order for you to receive information from the rods. And in fact, many different rods are connected to the same nerve fiber. So that means that even if each individual rod is picking up a very small amount of light energy, it will accumulate in that nerve fiber. What rods will not do well, and in fact you can generalize and just say they don't associate themselves with color. Okay, so rods are excellent at picking up information at very low intensity of light, but they're not good on detail, and they do not pick up color well. Now this is referred to as scotopic vision. Vision in which the rods are the main detectors of the incident light. So scotopic vision, which is usually referring to light of low intensity. Cones, on the other hand, they do distinguish different colours. So the cones are responsible for how we see in colour. There are 3 types of cone, each sensitive to a different colour, and fewer of them are connected to the same fibre. The consequence of this is that you can pick up way more information and have a lot more detail in the image. Cones are only sensitive when the intensity of light is high. This is why in low intensity light you can't see colours, because you're only using your rods in those kinds of situations. The definition then of photopic vision is vision in which the cones are the main detectors of the incident light. So you have scotopic and now photopic. So what do you have to do? You literally just have to learn all that information off by heart. And it's actually the most common question in paper 3. So if you have a look at the differences, scotopic and photopic. Scotopic vision is where the rods are being used. It tends to happen in situations where there's little light available, especially at night. You can distinguish shapes, but not colours, and you can't really see things with a lot of detail. So basically you just switch the lights off and sit in a slightly dark room, not a completely pitch black room, but a dark room, and you can see, have a look and think about what you're seeing. There's very little colour, there's very little detail. You're using your rods and they don't pick up that level of detail. Now, photopic vision, on the other hand, is when you're using your cones. Has to be when there is a lot of light available, so like the day, and then you can distinguish shapes and colours, and you should be able to distinguish a lot of detail. Now these are 2 extreme situations. What happens normally is that you're using both scotopic and photopic vision. The next thing that you need to know about is the distribution of the cones and the rods at the back of the retina. For this, I recommend seeing the video that I've put on Moodle, which is from Khan Academy, that explains it really well. However, I will go through it now. If we squeeze this out of the way, okay, so we can have a look. The part of the eye with the highest concentration of cones is the back of the retina, which has that dip in the fovea. Okay, so what you actually have is a huge concentration there of cones, which skyrockets up there. Okay, and that's why when things focus at the fovea, you see in such impressive detail and with lots of colour. And interestingly enough, that's where you have the least rods. The rod distribution actually increases as the cones decrease, and they go up like this and then come down. So if we have a look at the picture, what we can see is at the fovea we have the maximum amount of cones, and then as we move further out away from the fovea, we increase the amount of rods. And there's a very common question, which is why can you see around in the dark? If you think about it, you're squinting around in the dark. It's actually clearer at the sides of your vision. So you have to look out the corner of your eyes in order to see very clearly. And that's because that's at that sort of light, what we're dealing with is scotopic vision. The rods, they're kind of around the sides of your eyeball, so you kind of have to look out the corner of the eye because you're using rods, and they're the ones that are picking up the most detail. Now, how do the cones work in order to enable us to see the whole array of colours that we do? Well, there are 3 types of cone. Okay, we have one type which responds to the blue, predominantly blue wavelengths. Okay, one type that responds mainly to green, and one that responds to red. Now we'll talk a little bit later when we look at colour addition. We'll talk about why we only need 3 colours in order to produce all the ranges that we see, and we'll talk about that a little bit later on. But for now, let's have a look at this spectral response graph. Okay, I'm going to write it there, spectral response curve. You have 3 waves. So this is how the cones that respond to the blue wavelengths, how do they respond at these ranges? Now, as you can see, there is one peak wavelength which gives maximum response, but we have a reduction in response, and that would be the generation of an electrical signal in response to this wavelength. Okay, so you would have a much stronger electrical signal at 420. You don't really need to go into that in too much detail because the graphs themselves tend to be in arbitrary units. Now, if one of the cone types responds to blue, then you would get a different cone type 2, responding to all of these wavelengths, but peaking at 530, which is specifically the colour green. And then the last cone type, it reacts in the following way, increasing its reaction until it gets to 560, which is the colour red. And then it reduces. Now again, these aren't all bundled up into a few nerve fibres, they're actually spread out, so they can carry that information much more clearly. But this is a spectral response graph, and these are how the 3 cone types work. The information from each individual type, from the green, the blue, and the red, combines then, before it gets to the brain, to give the whole range of different colours. But we need to talk about colour addition. Now, I recommend watching the videos first, and then coming back to read through the text. So how do colours combine? Now, coloured light is not like the colours that you use in the art room. It's important that you've watched the videos because then you can see how the colour is added to create completely new situations. In an art room, if you put red, blue and green together, you get a messy brown. But what you get when you combine the 3 primary colours is pure white. Okay, and it's important that you see that. But let's revise primary colours. Primary colours are colours which, when overlapped, give the whole range of all the other colours. Okay, and in fact, adding the 3 primary colours together, we get white light. Okay, and what are those 3 primary colours? Blue, green, and red. Okay, so those are the 3 primary colours. Now, when you add those 3 primary colours together, depending on what combination, you get something called secondary colours. The 3 secondary colours are cyan, magenta, and yellow. And you need to know the combinations, unfortunately. Blue and green give you cyan, blue and red give you magenta, and red and green give you yellow. Again, it's best to see this on the video than just like learning it as if it was just text. It would be great if you could see the colours. The next layer is what happens if you then add further colours. Well, depending on the combinations, you could go back to white light. So cyan, which is actually blue and green, plus red, well, that's the 3 primary colours. So that gives you white. Magenta, which is actually red, and blue plus green gives you white. And yellow, which is actually red, and green plus blue give you white again. In the same way that you add colours, you can subtract colours too. And the way that you subtract colours is using pure filters. If you use a yellow filter, for example, and white light, what the filter does is absorb all the colours except for the one that it shows. So a yellow filter will absorb everything except for yellow. And that's why they work. That's why you can see yellow through a yellow filter. It becomes a bit more interesting when you use secondary colours. So we'll look at the magenta filter and the cyan filter. If we use a magenta filter, if you remember, magenta has blue and red. So white light, which has blue, red and green, will allow through blue and red and absorb all the rest. Cyan, which has got blue and green, will allow blue and green. Now that's easy enough, but we're going to look at what happens when you have two filters. If I first use a magenta filter, again, white light flows through, and magenta has red and blue. Okay, make that a bit neater. Red. So you'll get red and blue. But what happens if I put a blue filter here? Well, you will get red light coming through. If instead of using a red filter though, what would happen if I was to use a green filter? Well, red would be absorbed and blue would be absorbed, and you would end up with no light at all. It would be completely dark.